Hello students, sexual reproduction in flowering plants part 3. Today we are going to study pistil, megasporangium and embryo sac. As we know, female part of the flower is called pistil. Carpel or gynoecium. It contains three parts: stigma, style, ovary. Stigma is the platform to reach, to land pollen grain, and the stalk-like structure that you can see style. Then the bottom swollen part, ovary. Inside the ovary, ovule, placenta, egg is located. And if one flower, one pistil, then it is called monocarpellary. In one flower, more than one carpel or more than one pistil, then you can say multicarpellary. This more than one carpel, if it is fused, then it is called syncarpus. If it is free, then it is called apocarpus. Then there are three parts. As we know, we have studied stigma, style, ovary. Inside the ovary, ovarian cavity, or you can say locule. Inside the ovarian cavity, placenta is present. Then the number of ovules in ovary, that is different. If one ovary, one ovule, then you can study the example wheat, paddy, mango. Then if one ovary, there are more than one ovule, then the example you can understand papaya, watermelon, orchids. Why I am saying these examples are very important for the NEET. Next we can study structure of ovule or megasporangium. So megasporangium, or you can say ovule. Ovule is a small structure attached to the placenta by means of stalk. It's a small structure attached to the placenta by means of the small structure stalk is called funicle. The body of the ovule fuses with the funicle in the region. Funicle is fused. That region you can say hilum, and it is surrounded by the layer that you can say outer integument and inner integument. Inside the layer integument, inside the mass of tissue that you can say nucellus. Inside the nucellus, embryo sac is present. Now the nucellus, it is used to nourish the embryo sac. The mass of tissue that only you can say nucellus. This nucellus is nourish the embryo. And nucellus contain more than one nucleus, and it store the food. It is used to nourish the embryo. And here you can see embryo sac. Embryo sac is called female gametophyte. And here you can understand the word megasporogenesis. So the megaspore formed from MMC, that you can say megaspore mother cell. Megaspore mother cell, that also you can understand the ploidy of MMC is diploid 2N. So MMC undergoes meiosis to form megaspore tetrad. In 4 cells are formed, that only you can understand megaspore tetrad. Out of these 4 cells, 1 will be functional, remaining 3 will be degenerated. So only 1 will be functional, that only you can say functional megaspore, that only you can say female gametophyte. It is called embryo sac. Here you can see megaspore tetrad, 4 cells, that only it is called megaspore tetrad. Out of this 4 cell, 1 will be functional, that you can say functional megaspore, that only you can say embryo sac or female gametophyte. So the method of embryo sac formation from a single megaspore, that you can say monosporic development. So now one cell, one nucleus. This one nucleus undergoes mitosis to form 2 cell. Again the 2 cell undergoes mitosis to form 4 celled stage, means 4 nucleus. Nucleus only, here you can see the nuclear division. So 2 nuclear stage, and next you can see 4 nucleate stage. Again this 4 nucleate undergoes mitosis to form 8 nucleus. Out of this 8 nucleus, first 6 cells, 6 nucleus, it's covered by the cell wall. Remaining 2 together, 2 nuclei together covered by 1 cell. So at last you can see this is the structure of embryo sac or a female gametophyte embryo sac. So, 3 cells at the chelacal end, 3 cells at the micropylar end, 1 cell, 2 nucleus, that is at the center. So, here you can understand 3, here 1, 7 celled 8 nucleate stage, that only you can say embryo sac. So, 3 cells are present at the chelacal end is called antipodal cells. 3 cells present at the micropylar end, it is called egg apparatus. Out of these 3 egg apparatus, 1 cell is called egg, another 2 is called synergids. In the synergids, there is a finger-like structure that you can say filiform apparatus. Filiform apparatus is used to guide the pollen grain. Then at the center, there is a central cell or a large cell that you can say polar nuclei. So, this is the structure of embryo sac. Thank you, students.